So today we are going to start a new topic, that is antimycobacterial agents. Antimycobacterial agents, also called as the antitubercular agents. So this tuberculosis is caused by the bacteria or caused by the organism Mycobacterium tuberculosis, which is the causative organism of the tuberculosis. Which is the causative organism of the tuberculosis? So it is Mycobacterium tuberculosis. So by today, one third to one half of the world population is infected with the Mycobacterium tuberculosis, and around 6%, that is around 5 million of the death worldwide occur because of this tuberculosis. The tuberculosis occur because of the urban crowding, homelessness, immigration, drug abuse, then the disappearance of preventive medicine health clinics, then the crowded prisons, and the AIDS epidemic. So, because of these reasons, the tuberculosis caused to the persons. So, this tuberculosis or this organism, Mycobacterium tuberculosis, it is transmitted primarily via the respiratory route. The organism Mycobacterium tuberculosis, it appears in water droplets. It appears in the water droplets and expelled during coughing, sneezing, or talking. Means what? That is those persons who is suffering from the tuberculosis, and if that person come in the crowded area and he or she, that person, if he cough or if he sneeze or if he talk with the other person which is close in contact with him, then that person will also suffer from the tuberculosis. So either in the form of the droplet or as airborne bacilli, the organism enter the respiratory tract. The infectiousness, the concentration of this tuberculosis, it is totally depend on the extent of the disease as well as the number of organism in the sputum and the amount of coughing. So generally, if you see the person who is suffering from the tuberculosis, he cough randomly, repeatedly. So the coughing is the main sign of the tuberculosis. Continuous coughing is the main sign of the tuberculosis. Tuberculosis generally affect the lung. That is around 80 to 85% cases of the pulmonary tuberculosis are there. But the organism Mycobacterium tuberculosis, it is spread through the bloodstream and the lymphatic system to the brain, bones, eyes, and skin. So that tuberculosis is called as the extrapulmonary tuberculosis. That is, tuberculosis occur to the other organs except the lungs. That is, the tuberculosis of the lungs is called as pulmonary tuberculosis, and the tuberculosis is caused to the other organs except the lungs, it is called as the extrapulmonary tuberculosis. In pulmonary tuberculosis, the bacilli reach the alveoli and are ingested by pulmonary macrophages. The substances which is secreted by the macrophages, it stimulate surrounding fibroblast to enclose the infection site, leading to the formation of granulomas or the tubercles. So this is what all about the tuberculosis, introduction of the tuberculosis. Now we will see the drug therapy for the tuberculosis. Now regarding the drug therapy of tuberculosis, these are classified into the two major classes. The first one is a first line agent and the another one is the second line agent. First line agents and the second line agents. So depend on the name. If you see the name, first line agent means what? That is the tuberculosis disease. Those persons who just started with the disease of the tuberculosis. So to those persons, first line agent has given. Those persons who enter the developing stage of the tuberculosis, that is second stage or third stage of the tuberculosis. So to those patients, either the combination of first line agent and the second line agent, both the agents are given in combination, or something different drugs they are given. So likewise, we will study the different agents of the tuberculosis. Again, these tuberculosis agents develop a rapid resistance. They develop the rapid resistance. Hence, those persons who is suffering from the tuberculosis and those who develop resistance against the first line agent, so to those patients, the second line agents has given. So, the disadvantage or the drawback of these tuberculotic drugs is what? That is, they develop the rapid resistance against the agents. So we'll see the drugs one by one. So the first class is the first line agents. So in this class, we're going to see 6 drugs in first line agents. So the first drug is the isoniazid, which comes under the first line agents of the tuberculosis. So this is a simple structure of the isoniazid. It is also called as INH in short form. Isoniazid, it is called as INH. So this is a simple and straight structure of the isoniazid, as it contains the pyridine ring, and to this pyridine ring there is a hydrazine group and the carbonyl group. So the IUPAC nomenclature of the isoniazid is pyridine 4 carbohydrazide. Pyridine 4 carbohydrazide. So this is the simple IUPAC nomenclature of the isoniazid. Now this isoniazid or the INH, it is developed or it is synthesized in year 1950s as a beneficial agent effective against intracellular and extracellular bacilli, and it is generally considered to be the primary drug of treatment of the Mycobacterium tuberculosis, as this is the first drug of the first line agent. So initially the patients are given with the isoniazid drug, those who suffer from the tuberculosis. Regarding the mechanism of action, this isoniazid drug inhibit the enzyme mycolase synthetase. Mycolase synthetase, the enzyme necessary for the biosynthesis of the mycolic acid. As the name of the enzyme suggests, the name of the precursor. That is, if we inhibit the enzyme mycolase synthetase, then it inhibits the biosynthesis of the mycolic acid. The latter, that is the mycolic acid, are important constituent of the mycobacterial cell wall, since mycolic acid are present only in the mycobacteria. So hence the mycolic acid is not available for the synthesis of the mycobacterial cell wall, and then the cell is dies. So what is the mechanism of action of the isoniazid? That is, this agent isoniazid, they inhibit the enzyme mycolase synthetase, and thereby they inhibit the biosynthesis of mycolic acid, and this mycolic acid is an important precursor of the mycobacterial cell wall. So mycolic acid is not available for the for the biosynthesis of the bacterial cell wall, then the cell wall or then the cell is dies. So this is what the isoniazid. Regarding the structural activity relationship of the isoniazid, so this is the basic structure of the isoniazid. So there are few points of the SAR of the structure activity relationship. That is, the N1 nitrogen in hydrazine side chain should be unsubstituted. So this one is the N1 nitrogen. That N1 nitrogen should be unsubstituted. Then the N2 nitrogen can be substituted with alkyl groups to get the active compounds. Then the third one, replacement of the pyridine nucleus with other aromatic rings such as benzene or piperidine or thiazide ring diminish the antitubercular activity. And the last one, numerous derivatives have been developed. None of them have exhibited activity superior to that of the parent drug, that is to that of the isoniazid. So these are the few SR points of the isoniazid. So what is the points? The first one, that is an N1 nitrogen should be unsubstituted. This nitrogen should be unsubstituted. Then N2 nitrogen, it should be substituted with alkyl group to get the active compounds. If we replace this pyridine ring with the other aromatic ring, then the activity will decrease. And there are various derivatives are developed, but no one of the agent will show the activity when we compare it with the isoniazid. So this is about the SR. Then these are the metabolic product of the isoniazid. So this is the isoniazid and these are the various metabolic product. So the first and primary metabolic product is the N-acetyl isoniazid. The first metabolic product of the isoniazid is the N-acetyl isoniazid. The enzyme responsible for the acetylation is the systolic N-acetyl transferase. It is that enzyme is present in the liver and in the small intestine. So this metabolic product, that is the N-acetyl isoniazid, it is observed in the liver as well as in the small intestine. And the enzyme is the N-acetyl transferase. N-acetyl transferase. The second major metabolic product of isoniazid is the isonicotinic acid. It is found in the urine. It is found in the urine as conjugate, as glucuronic acid conjugate or the sulfate conjugate. And it is formed by hydrolysis of N-acetyl isoniazid. Okay, this isonicotinic acid, it is formed by the hydrolysis of N-acetyl. So this is the N-acetyl isoniazid, and the hydrolysis of N-acetyl isoniazid results in the formation of the isonicotinic acid. And this metabolite is found in the urine as the conjugate with the glucuronic acid or with the this sulfate conjugate. Then there are several other metabolites are formed like the acetyl hydrazide, then the diacetyl hydrazide, then the hydrazine. Then these two are, again are the metabolic products of the isoniazid. So this is what in short the regarding the isoniazid. Okay, now the next drug of the first line agent is the rifampin. The rifampin. Now, what actually is the rifampin? So this is the very bigger structure of rifamycin SV, and this is the rifamycin SV, and this is the general structure. If in this case, if R is CH3, then it is rifampin, and if it is cyclopentyl ring, then it is rifapentin. Okay, so this is the structure of the rifamycin SV, and this is the structure of the rifampin and the rifapentin. There are substitution of R. So the rifamycins are members of ansamycin class of the natural products produced by Streptomyces mediterranae. So these rifamycins are isolated from Streptomyces mediterranae organism. This chemical class is characterized as molecules with an aliphatic chain. So these are the aliphatic chain forming a bridge between two non-adjacent positions of the aromatic moiety. So these are the two aromatic rings which are fused with each other. So it forms a bridge between these two aromatic rings. The rifamycins, they inhibit. So this is the mechanism of action of the rifamycin or the rifampin. So the rifamycins, they inhibit bacterial DNA-dependent RNA polymerase enzyme by binding to the beta subunit of the enzyme. So this rifamycin bind to the beta subunit of the DNA-dependent RNA polymerase enzyme, and it is highly active against rapidly dividing intracellular and extracellular bacilli. So the inhibition of the DNA-dependent RNA polymerase lead to blocking the initiation of chain formation in RNA synthesis. What happen if we inhibit this enzyme? What happen if we inhibit the DNA-dependent RNA polymerase enzyme? It lead to blocking the initiation of chain formation in RNA synthesis. That is what the RNA synthesis, it is get inhibited. RNA synthesis is get inhibited as the chain will not elongate. The RNA chain will not elongate. So that is what the mechanism of the action of the your rifamycin. So the DNA-dependent RNA polymerase, it is a metalloenzyme. It contains the metal ions, which contain two zinc atoms. As it is a metal enzyme, so it contains a two zinc atom. It is further postulated that the oxygen at carbon number 1 and carbon number 8, so where it is. Okay, so if you check, so this is the carbon number 1, hydroxy oxygen, and the carbon number 8. So this is the hydroxy oxygen. So 1 and 8. So here they are. So it is postulated that the oxygen at carbon number 1 and carbon number 8 of rifamycin can chelate to zinc atom. So where are the zinc atoms? Zinc atoms are present at the enzyme as it is a metal enzyme. So the oxygen atom which is present at carbon number 1 and carbon number 8 of rifamycin, they chelate with the zinc atom, which increases the binding to the DDRP, that is the DNA dependent RNA polymerase enzyme. And finally, the oxygens at carbon number 21 and carbon number 23, they form strong hydrogen bond to the DDRP. So this is how they bind and how they inhibit the DNA dependent RNA polymerase enzyme. So what actually is the mechanism of action of the rifamycin is, that is this DNA dependent RNA polymerase enzyme, it is a metalloenzyme, that is it contains the zinc atoms, whereas our molecule, that is rifamycin, it contains oxygen atom at carbon number 1, carbon number 8, carbon number 21 and carbon number 23. Okay, so at carbon number 1 and carbon number 8 of the rifamycin, they bind to this enzyme by chelating with the zinc atom, whereas the oxygen which is present at carbon number 21 and carbon number 23, they form the strong hydrogen bond with the DDRP. So thereby there is a strong interaction of the drug with the DDRP enzyme, and thereby they inhibit the enzyme. Once they strongly bind to the enzyme, they inhibit the enzyme, and thereby they block the RNA synthesis of that particular cell. So this is what the mechanism of action of the rifamycins. Regarding the structural activity relationship, so of course, that is hydroxy group is essential at carbon number 1, 8, 21 and 23 because they are involved in the binding of the enzyme. Then these groups appear to lie in a plane and to be important binding group for attachment to DDRP. Yes, definitely. That groups, that is the hydroxy group which is present at 1, 8, 21 and 23, they lie in a same plane for proper binding to the enzyme. Then acetylation of carbon number 21 and carbon number 23 produces inactive compound. If we acetylate the carbon number 21 and carbon number 23, this one 21 and 23, 21 hydroxy and 23 hydroxy. So if we acetylate this group, it produces the inactive compounds. Reduction of the double bonds in the macro ring results in a progressive decrease in activity. So if you reduce the double bonds, over here, there are lots of double bonds in the structure. So if you reduce that double bonds, then it results in the loss of the activity or the decrease in the activity. And opening of the macro ring also gives the inactive compound. If we open this ring, now this is a flow structure. So if we open the ring, then it results in the loss of the activity, or it results in the inactive compound. These latter two changes, that is the point number 4 and point number 5, that is the reduction of the double bond and the opening of the ring. So these latter two changes greatly affect the conformation structure of the rifamycins, which in turn decreases binding to the DDRP, that is a DNA dependent RNA polymerase enzyme. Then the next point, substitution at carbon number 3 or carbon number 4. So here it is, carbon number 3, this one, and carbon number 4, this one hydroxy. So the substitution at carbon 3 or carbon 4 results in compound with varying degrees of antibacterial activity. The substitution at this position appear to affect transport across the bacterial cell wall. So if we substitute at this position with any other groups, then it affect the transport across the bacterial cell wall. Then these are the metabolites of the rifampin. So rifampin, by deacetylation, It is results in the formation of the diacetyl rifampin or the diacetyl rifampicin. Both are the important drugs in the treatment of the tuberculosis. If R is CH3, then it is rifampin, and if it is C5H9, that is if it is cyclopentyl ring, then it is rifampicin. Then rifampin by oxidation also results in the formation of this metabolic product, and again rifampin by hydrolysis results in the formation of this 3-formyl rifamycin SV. So this is what the metabolism of the rifampin. So rifampin undergo deacetylation, undergo hydrolysis, and undergo oxidation to form various products. So we have studied about the rifamycin and the rifampin. Now the next one is the rifampicin. As we see the structure of the rifampicin. Okay, so this is the structure of the rifampicin. It contains the cyclopentyl ring. So rifampicin. Rifampicin is the first new agent introduced for the treatment of the pulmonary tuberculosis in the last 25 years. The drug's major advantage over rifampin, RIF means what? It is rifampin. RIF is what? Rifampin. So the drug's major advantage over rifampin is the fact that when used in combination therapy, rifampicin can be administered twice weekly during the intense phase of therapy, followed by once a week administration during the continuous phase. So the dosing is very less. So what is the dosing? If we compare the dosing of the rifampicin with the rifampin, so what is the dosing of the rifampicin? That is twice weekly during the intense phase of therapy, in a week 2 times. Then after few weeks, once a week administration during the continuous phase. So this is the dosing of the rifampicin. Now in contrast, rifampin normally is administered daily during the intense phase. So here is the difference. In the intense phase, rifampicin twice weekly. In the intense phase, rifampin administered daily, followed by twice a week during the continuous phase. In the continuous phase of the tuberculosis, rifampicin gives once a week. Over here, it is given twice a week. So this is the difference of the dosing regimen. So rifampicin is more potent as compared to the rifampin. Initial clinical studies actually showed that the relapse rate in patients treated with rifampicin 10% were higher than those in patients treated with the rifampin. So the rifampicin drug is more potent as compared to the rifampin. Then the next agent. So we have studied the rifampicin, we have studied the rifampin, we have studied the isoniazid. Now we'll study the next drug, that is the pyrazinamide. Okay, so the next drug is the pyrazinamide. So what actually pyrazinamide is? Pyrazinamide, pyrazinamide, also called as the pyrazine carboxamide, it was discovered while investigating analogs of the nicotinamide. Pyrazinamide is a biester of the nicotinamide and possesses bactericidal action against Mycobacterium tuberculosis. So the name suggests the structure of the pyrazinamide. That is, it contains pyrazine and the amide group. So the IUPAC nomenclature is what? Pyrazine 2-carboxamide. Regarding the mechanism of action of the pyrazinamide, as it is unclear, Mycobacterium tuberculosis has the enzyme pyrazinamidase, which is active in the acidic conditions. So this organism, the causative organism of the tuberculosis, that is the Mycobacterium tuberculosis, it has the enzyme pyrazinamidase, which is active in the acidic conditions. So this pyrazinamide is converted in pyrazinoic acid by the enzyme pyrazinamidase. So as the drug is used against the Mycobacterium tuberculosis, but the Mycobacterium tuberculosis has the enzyme called as a parazinamidase. So what this enzyme do? This enzyme convert our drug into the pyrazinic acid. This pyrazinic acid inhibit enzyme fatty acid synthase 1, which is required by the bacteria to synthesize the fatty acids. So our drug, that is our parazinamide drug, it is a prodrug. It is get converted into the pyrazinoic acid by the enzyme pyrazinamidase, and this enzyme is part of the Mycobacterium tuberculosis. So this enzyme parazinamidase, it converts parazinamide into the parazinic acid. Once the parazinic acid is get formed, it inhibit another enzyme called as a fatty acid synthase 1, which is required by the bacteria to synthesize this fatty acid. So fatty acid will not available for the bacteria to synthesize its bacterial cell wall. Hence, the cell will dies. Okay, so this is what the mechanism of action of the parazinamide. Regarding the structural activity relationship, substitution on the pyrazine ring or use of alternate heterocyclic aromatic ring has given compounds with reduced activity. So if we substitute the pyrazine ring with any other heterocyclic ring, it gives compound with the reduced activity. More recently, using QSAR, a series of analogues have been prepared with improved biological activity. The requirement for successful analogues include: first point, that is provision for hydrophilicity to allow sufficient plasma concentration such that the drug can be delivered to the site of action. That is, that the drug should have the hydrophilic group so that the drug can be delivered at its site of the action. Again, the drug is having a lipophilic group to allow penetration into the bacterial cell wall. Third point, the drug should undergo hydrolysis reaction. So these are the 3 points which are taken into the consideration while developing the analogues of the parazinamide. So two compounds have been found that meet all these criteria. The one is the tertiary butyl 5-chloropyrazinamide. The second one is the 2-2-methyl decyl 5-chloropyrazinamide. So these are the two drugs which are synthesized by taken into the consideration of all these 3 points. And these two drugs are active as a parazinamide. So this is how the parazinamide is get converted into the parazinic acid, and the parazinic acid is further converted into the 5-hydroxy parazinic acid. So these are the metabolites of the parazinamide. So the parazinamide is get converted into the parazinic acid by the enzyme parazinamidase, which is the enzyme of the Mycobacterium tuberculosis. Further, the parazinic acid is get converted into the 5-hydroxy parazinic acid.
The next drug is ethambutol. As you know, this is the structure of the ethambutol. Now, this structure contain alcoholic functional group. Now, in this case, this is the butanol. In this case, the functional group is butanol. So, we'll start the aggregation nomenclature from this butanol. So, this is a straight chain 1, 2, 3, 4. 4 carbons in a straight chain. So, it is a butanol. Butan 1-ol. So there is a substituents get attached at the second carbon of the butanol. So we'll start with the 2- of the butanol. So 2, then again 2, 1, 2, then 1-hydroxy butan 2-yl amino ethyl amino. The remaining portion, that is a butan 1-ol. So this is the IPC nomenclature of the ethambutol. Now ethambutol, this drug inhibit the enzyme arabinosyl transferases, This enzyme is involved in the biosynthesis of the cell wall. So once we inhibit this enzyme, carabinosyl transferase, then the bacterial cell wall synthesis is get further inhibited. Regarding the structural activity relationship, that is the number of studies are carried out on the ethambutol to emphasize its activity. So the SR studies indicated that the nature of branching, the nature of this kind of branching, distance between the two nitrogen atoms, and the extent of N-alkylation. These are the activity governing factors. These are some metabolites of the ethambutol. That is, ethambutol undergo a reduction to form this metabolite A, and by oxidation to form metabolite B. Then the next drug is streptomycin. So this is the last drug of the first line agent, streptomycin. Now streptomycin, it is, we have already studied in the antibiotic chapter in the aminoglycoside antibiotic class. So this streptomycin was first isolated by the Weissman and co-workers in 1944. Regarding the mechanism of action, the streptomycin act by binding to the bacterial 30S ribosomal subunit, which cause misreading of the tRNA, and it leaves bacteria unable to synthesize protein. The proteins are required for the growth. Regarding the SR, that is, several modifications are carried out on the alpha streptose portion. So this is the alpha streptose portion. So the several modifications were carried out on this alpha streptose ring to get better compound. So which kind of modification? So the first one, they reduce aldehyde to the alcohol. So this is the aldehyde group on the alpha streptose ring. So they reduce this aldehyde group to the alcohol, which results in a formation of the compound dihydrostreptomycin, which has activity similar to streptomycin, but with greater potential for producing delayed severe deafness. Then also they carried out oxidation of these aldehyde group to the carboxyl group. Oxidation of this aldehyde group to the carboxyl group, or conversion to the shift-based derivative like oxime, semicarbazone, or phenylhydrazone, which results in the formation of the inactive analog. Then oxidation of methyl group. So over here, oxidation of methyl group in alpha streptose to a methylene hydroxy, it gives an active analog that has no advantage over streptomycin. Then modification amino ethyl group. So this group, this is a glucosamine portion. So the modification of the amino methyl group on the glucosamine portion of the molecule by demethylation or by replacement with larger alkyl group reduces the activity. And at last, that is the removal of this guanidine ring. It decreases the activity of the streptomycin. So this is what all about the first line antimycobacterial agents. Okay, so the next class is the second line agents. So the first drug of the second line agent is ethionamide. Now this is the structure of the ethionamide. This ethionamide structure contains pyridine ring. To this pyridine ring, there is a substituent at second position. So start the IUPAC numerator from this substitute because we have to follow the alphabetical order. So it is 2-ethyl 4-carbothioamide. The synthesis of analogs of isonicotinamide results in the discovery of ethionamide. and a homologue in which the ethyl group is replaced with a propyl, which results in the formation of the prothionamide. Now, both the compounds, that is the ethionamide and the prothionamide, they have proven to be bactericidal against the Mycobacterium tuberculosis and the Mycobacterium leprae. And this ethionamide drug is having the same mechanism of action as the isoniazid. That is, this drug inhibits the enzyme mycolase synthetase. This enzyme is necessary for the biosynthesis of the mycolic acids, and this mycolic acid is the important constituent of the mycobacterial cell wall. So these are the metabolites of the ethionamide. Now these ethionamides are get metabolized into the ethionamide sulfoxide, then the 2-ethyl isonicotinamide, and various other compounds. So these some are the metabolites of the ethionamide. Now the second drug is the para-aminosalicylic acid. The IBC raw temperature is, now this drug contains the carboxylic acid as a functional group. So we have to start the numbering from the carboxylic acid. So as per alphabetical order, we have to start the numbering from this fourth position. There is a para position of the carboxylic acid group. So 4-amino 2-hydroxy benzoic acid. Now this para-aminosalicylic acid is thought to act as an antimetabolite, interfering with the incorporation of PABA, that is the para-aminobenzoic acid, into the folic acid. When co-administered with the isoniazid, this para-aminosalicylic acid is found to reduce the acetylation of the isoniazid, itself being the substrate for acetylation, thus increasing the plasma level of the isoniazid. In short, this para-aminosalicylic acid, when given with the isoniazid, so this para-aminosalicylic acid delay or inhibit the metabolism, that is the acetylation of the isoniazid, and thereby the isoniazid is available for the more time in the plasma and give its action. Now this action can be especially valuable in patients who are rapid acetylators. This para-aminosalicylic acid is extensively metabolized by acetylation of the amino group. So over here, the acetylation takes place. That is results in the formation of the metabolite NHC-OCH3, and by conjugation with the glucuronic acid and glycine at the carboxyl group. So that is a phase 2 metabolism. That is a conjugation with the glucuronic acid and glycine at the carboxyl group. Then the next drug is the cycloserine. Now this is the structure of the cycloserine regarding the IBC raw temperature. So this ring is the oxazolidine ring, 1,2-oxazolidine ring. So regarding the IBC raw temperature, there is an amino group which is present at the fourth position of this oxazolidine ring. So we start with the alphabetical order. So 1, 2, 3, 4. So it is 4-amino, 1,2-oxazolidine, 3-on. Now this cycloserine, it is isolated from Streptomyces auricadiceus as a dextroenantiomer. So this dextrocycloserine, it is the active form of the drug and having its action associated with the ability to inhibit the two key enzymes, that is the D-alanine racemase and the D-alanine ligase. So this D-cycloserine inhibit the two enzymes, that is the D-alanine racemase and the D-alanine ligase. Now this D-alanine, it is an important precursor, or it is an important constituent of the peptidoglycan layer of the bacterial cell wall. Now these mycobacteria are capable of using naturally occurring L-alanine, and it converts this L-alanine to the D-alanine via enzyme D-alanine racemase. And the resulting D-alanine, which is formed from the L-alanine from the mycobacteria, that D-alanine is coupled with the another D-alanine molecule to form D-alanine complex under the influence of the enzyme D-alanine ligase. And then this complex, that is a D-alanine complex, it is incorporated into the peptidoglycan layer of the mycobacterial cell wall. Now, the D-cycloserine is coming to the focus. Now, this D-cycloserine is having the structural similarity with the D-alanine, and thereby this D-cycloserine compete with the D-alanine for the binding to the active site of the enzyme or where the D-alanine bind, and thereby the D-cycloserine inhibit the binding of the D-alanine to the active site of the enzyme. Then the next drug is the capreomycin. So this is the structure of the capreomycin. Now this capreomycin is a mixture of 4 cyclic polypeptides, of which capreomycin 1A and capreomycin 1B make up 90% of the mixture. This capreomycin is produced by Streptomyces capreolus. The mechanism of action is not known, but it is thought to inhibit the protein synthesis by binding to the 70S ribosomal subunit. Like streptomycin bind to the 30S ribosomal subunit and thereby they inhibit the protein synthesis. Now this capreomycin, they bind to the 70S ribosomal subunit and inhibit the protein synthesis. This capreomycin also bind to the components in the bacterial cell wall, which results in the production of abnormal proteins. These proteins are necessary for the bacteria's survival. Therefore, the production of this abnormal protein is ultimately fatal to the bacteria. Then the next drug is the fluoroquinolones. Now these fluoroquinolones, they are the broad spectrum class of the antibacterials and have to demonstrate the activity against the wide range of gram-positive and the gram-negative pathogens. The mechanism of action of the fluoroquinolones involve binding to the DNA gyrase DNA complex, which inhibit the bacterial DNA replication and the transcription process. As a result, this drug exhibit bactericidal activity. Now these are the fluoroquinolones agents. Now those quinolones which doesn't have the fluorine ring, these drugs are inactive against the mycobacteria. In addition, it has been reported that certain fragments or substructures within the quinolones improved activity towards the MAC, whereas other fragments deactivate the quinolones. Now there are some important structural features of the fluoroquinolones, and that features include that is a cyclopropyl ring at the N1 position. So this is the N1 position. So there should be a cyclopropyl ring at the N1 position. Then the fluorine atom at the C6 position and the C8 position. Then a C7 heterocyclic substituent should be present. Then the excessive lipophilicity at N1 can decrease the fluoroquinolone activity. The example of such compound is 2,4-difluorobenzene. Then C7 substituents with greatest activity against mycobacteria include the substituted piperazine and the pyrrolidines. Several C8 methoxy substituted fluoroquinolones have been reported with the superior activity over earlier quinolones. The moxifloxacin is reported to be active against the Mycobacterium tuberculosis when it is combined with the isoniazid. And the last drug is the bedaquiline. Now this is the structure of the bedaquiline. Again, this drug contains the alcoholic functional group. Over here, it is butanol. 1, 2, 3, 4 years. The 4 carbons in a structure. 1, 2, 3, 4 years. So it is butane to all. So we start the episode ratio from the first position of the butanol, because we have to follow the alphabetical order. So as per alphabetical order, we will cover this bromine first. So 1 dash, then 6 bromo, 1, 2, 3, 4, 5, 6, 6 bromo, 2 methoxy, quinoline, 3YL, then 4 dash of this butanol, 4 dash, dimethyl amino, then 2 dash of this butanol, 2 dash naphthalene 1YL, naphthalene 1YL, 1 phenyl butane 2-ol. So this is the IPC temperature of the betacholine. Now this betacholine is generally given with the other drugs for the treatment of the tuberculosis. Means this betacholine is given in combination for the treatment of the tuberculosis. This betacholine, it is a diarylphenolene antimycobacterial drug which inhibit the proton pump of mycobacterial ATP synthase. This enzyme is essential for the generation of energy in the Mycobacterium tuberculosis. So once we inhibit this ATP synthase enzyme, the mycobacteria does not get energy. So the cell will die because of the lack of the energy. Now this bacterial death occurs as a result of the betacholine. Regarding the metabolite, that is an N-mono-desmethyl metabolite. So one of the alkyl group is liberated or it is get removed, results in the formation of the metabolite of the betacholine.